again, another eye-opening property for us that we wanted to delve further into and study further and understand the why. So this is kind of where Dr. Webster comes in, and we've been working with him for quite some time now, looking at mechanisms of these two properties and why they are what they are and how they're working. So with that, I'll turn it over to him.
Yeah, thank you, Jim, for that nice introduction about PEKK and how we've been collaborating with OPM to explore some more of its novel properties, as well as a why. Why does PEKK behave this way, improve bone growth, and inhibit bacteria functions? So Jim, if you want to go to the next slide. I'm at Northeastern University, as Jim mentioned, in chemical engineering. We have over a 25-year experience looking at novel materials in orthopedics, and that's how we got connected with OPM to do some studies on their materials. So next slide. Yeah, so this is just a description of some of our experience over that 25-year period, whether we have looked at 3D printed spinal implants or amputee devices, or in the bottom corner, looking at increasing bone growth on materials or increasing orthopedic soft tissue, like tendons or ligaments, or in the bottom left corner, looking at increased vascular endothelialization or decreased bacteria attachment on catheters. What we have found and what intrigued us about OPM's PEKK material is nanostructured surfaces can control cell responses. We have seen, a lot of examples of where nanostructured surfaces on different materials can reduce bacteria attachment, can inhibit inflammation, scar tissue growth, and ultimately can increase tissue growth, like bone growth, for example, as highlighted on this slide. So clearly PEKK is one of these very fascinating nanostructured materials that, personally, I wanted to get my hands on and test and see what kind of properties this new chemistry possesses. So next slide, Jim. So as an example for PEKK, here's some of our bacteria results that Jim was referring to before that we have published. We, like many groups, have been looking at non-antibiotic strategies to reduce bacteria colonization. And I think, as everybody knows, I know we're in a current pandemic with COVID-19, but the CDC has predicted further healthcare problems due to the emergence of antibiotic-resistant bacteria. In fact, by 2050, the CDC has predicted more people will die from antibiotic-resistant bacteria than all cancers combined, right? I mean, that's a startling statistic that should make us all think about developing surfaces of orthopedic implants that can reduce bacteria function without resorting to antibiotic use. So clearly, as we have seen, and I'll show some of these results here in terms of colony forming unit assays, that bacteria attachment and growth is much less inhibited on PEKK compared to PEKK, which is in blue on this slide, and titanium, which is in gray. And the y-axis on this slide are the number of colony forming units for one particular bacteria called Staphylococcus epidermidis. This is the bacteria on your skin that commonly infects orthopedic implants. And we have seen upwards of 7 days of reduction in Staphylococcus epidermidis colonization on PEKK, simply because of its chemistry, because of its nanostructured features, because of its inherent properties, not requiring an antibiotic or the release of an antibiotic. And these are in vitro results. So Jim, the next slide really tells the familiar story that we are seeing on these materials. Same graph outline, same color code for the materials where PEKK is in yellow, but here we're studying Pseudomonas aeruginosa, another type of bacteria that infects orthopedic implants, and we're seeing the same proactive bacteria inhibition upwards of 7 days in culture, again, without the use of an antibiotic. And I would also mention this result is incredibly promising to me as a researcher, because now we have looked at gram-positive and gram-negative bacteria. There are two types of bacteria that can infect your implant, and many times you need different antibiotics for those different bacteria. But here we have the same surface in PEKK that is reducing colonization of these two types of bacteria. So next. And the third type of bacteria that we have been looking at is MRSA. Of course, MRSA is the methicillin-resistant version of Staphylococcus aureus. So methicillin is an antibiotic, and this is a great example of why I think in the field we need to move beyond antibiotics as a way to treat orthopedic implant infections, because we created MRSA through our use of the antibiotic methicillin. And it's proven to be quite a hard bacteria to reduce colonization on an orthopedic implant, but the same exact results that we're seeing on PEKK as what I described for Pseudomonas and Staphylococci, a reduction on PEKK without the use of antibiotics upwards of 7 days in culture. So next, we just quickly summarize some of these results from the bacteria portion that we're seeing reduced bacteria colonization. We see the best response actually on MRSA, the antibiotic resistant version. So very promising properties. And now we've started looking into why. Why are these surfaces reducing bacteria colonization? So you can see on this slide, I've highlighted some reasons why nanotextured surfaces seem to be working in medical devices. In the upper left, you can see that natural structures like the insect wing, the surface of an insect wing has nanostructured features. So just emulating those nanostructured features in medical devices can improve an antibacterial response. On the upper right, we've done some work with finite element analysis, looking at that bacteria, which is red in that figure. How does the membrane adapt to nanostructured features? And what we find out is it doesn't adapt to nanostructured features. The bacteria membrane is a little too stiff to fit into those contours, and we don't get a good attachment of bacteria. So through biophysical models, you can explain this behavior. The last reason is shown right in the middle of the slide, and that is that these nanotextures have different surface energy, and that results in different protein adsorption to repel bacteria. So on nanostructured features, you have altered protein adsorption, altered protein bioactivity, which can keep an implant from being infected. So now, Jim, for the next slide, the specific results we're seeing on PEKK is when we measure its surface energy. As you can see in this table, the surface energy of PEKK is actually closer to 3 proteins in your body that have a natural ability to reduce bacteria colonization. Those 3 proteins, as you can see on the table, casein, mucin, and lubricin. If you can get these 3 proteins to adsorb to your implant, you can reduce bacteria colonization. So the chemistry of PEKK, the surface topography of PEKK, gives it a surface energy that's closer to these great proteins in your body that naturally resist bacteria colonization. So the next slide really shows where we've seen, in fact, increased adsorption of these proteins in vitro on PEKK compared to titanium and PEKK. In other words, PEKK has that surface energy to increase the adsorption of these proteins, and through ELISA assays that we've completed in the lab, we're seeing 5 times, 6 times more adsorption of these great proteins on PEKK compared to other materials. Next slide. And the last part of this bacteria story is we've correlated that increased adsorption of casein, mucin, and lubricin to decreased bacteria colonization. So we coated these materials, PEKK, titanium, and PEKK, with casein on this slide, and then we allowed Staphylococcus epidermidis to adsorb, and they're reduced. Same with Pseudomonas aeruginosa, correlating this increased adsorption of casein on PEKK to decrease Pseudomonas colonization. And the last one, MRSA, and all the other proteins and on the samples, we have correlated that increased adsorption to decrease bacteria colonization. So the summary on the next slide basically highlights this mechanism of action, that PEKK has the chemistry, has the roughness to absorb 3 key proteins in your body that are known to reduce bacteria colonization. That is the mechanism of action, not related to an antibiotic, not related to releasing reactive oxygen species or any other mechanism that people are looking for other materials. And the last part of what I wanted to share with you, Jim, the next slide, is this mechanism for greater osseointegration that Jim had talked about, this wonderful improved bone growth that you're seeing on PEKK compared to other materials. So the next slide is, we of course determined in vitro that more bone cells attach to PEKK. And if you get more cells to attach, then more cells will grow and then more bone will form. So clearly we're seeing greater amounts of bone cell attachment on PEKK compared to these other materials. The next slide, Jim, that is leading to greater mineralization. So a bone cell secretes calcium, secretes bone-related proteins, that is bone. And we call that mineralization, where again, you can see greater amounts of mineralization by osteoblasts, the formation of bone in vitro on PEKK compared to these other materials, PEKK and titanium. So the next slide is really, again, this question of why. Why is this happening? And we go back to the proteins because we see this unique surface energy of PEKK, which is matching the surface energy also of other proteins. And these proteins are known to improve bone cell response. So those proteins are vitronectin, fibronectin, laminin. These proteins are great for promoting bone cell attachment, which we saw, and greater bone formation, mineralization. So we're seeing PEKK has a greater tendency to absorb those proteins, as well as what I mentioned before, the antibacterial proteins, to create for a more antibacterial and more pro-bone growing surface. So this is, I think, is the last data slide that I'll show you that is correlating this increased adsorption of calcium that we're seeing, this increased protein adsorption of vitronectin, fibronectin, and laminin, key proteins in your blood that promote bone growth if they adsorb to an implant. And those osteoblast adhesion numbers are much greater when we pre-coat PEKK with these proteins and calcium. So again, correlating that if PEKK is absorbing more of those proteins, once they're absorbed, they're promoting bone cell attachment. So next is just the overall summary that we're seeing on PEKK materials when we look at these mechanisms of actions, that we're seeing greater initial bone cell attachment, greater bone cell growth in terms of mineralization and extracellular matrix synthesis. And it's related to the unique chemistry, surface roughness of PEKK that is absorbing key proteins in vitro immediately after implantation that is providing antibacterial and pro-bone growing properties. And I think that's it for our recent results, Jim.
Great. Thank you so much, Tom. It was great. So thank you, everybody, for attending the presentation.